Good morning and welcome, everyone. Thanks for joining us today for our monthly webinar series, and we're excited to have you here. My name is Zach Stoll, and I'll be your moderator for today's session. Just jumping right into the introductions, our speaker this month is Dr. Aaron Redman with ExxonMobil, and his talk today is titled Integrated Evaluation of Chemistry and Toxicity Data in Treated Produced Water. He brings a wealth of experience, previously working as a consulting engineer and is an active member in the research community, having published 72 papers across a range of disciplines in collaboration with industry, government agencies, and academic institutions. He studied at the University of Delaware, the Colorado School of Mines, whoop whoop, and Brigham Young University. Raised in the foothills of Colorado, Dr. Redmond has lived in Brazil, Brussels, and the northeastern US. Welcome, Dr. Redmond. The virtual stage is yours.
All right, awesome. Thank you. And again, I really appreciate you making the time and extending the invitation. Really excited to be part of this, put the presenter mode. Really excited to be part of the seminar series. I found it really useful conversation, like all the previous seminars. So really excited to have this as our contribution. Quick question. Do you see the presenter mode? The
Yes,
notes on.
we see your screen, but it's just still not in, it's not full screen yet. You still see the sidebar and everything.
Yeah, is that better?
All right. I don't know for, oh, there it goes. Yep, full screen.
All right, awesome. Okay, well, cool. So yeah, they asked like the folks in Mexico State and the Produced Water Consortium asked us to discuss one of our recent papers that just came out over earlier this summer, and really excited to be part of the seminar series and discuss. And I'm also really excited to see so many familiar faces and names on the discussion today. This is going to be great. Um, what I'm presenting on a page, like this is the list of all the co-authors on this specific paper, and I'll show you the link in a second. And what you're going to notice is that this specific piece of work is a collaboration with our friends over at the Colorado School of Mines. So James, Brett, and Zahi, and it also spans a couple different iterations of our produced water team here at ExxonMobil Biomedical Sciences. And so you'll notice familiar names like Josh Butler and Brian Hedgepeth, who used to be in this role a few years ago. So really excited to represent that work here. So we'll carry on. The second slide, Zach did a really nice summary, which is more or less what's on this slide. But one of the things, we've been very active in the research community over many years as a way to, leverage common data sets and to develop consensus positions or opinions or thoughts where they're available or where we can. And one of the approaches we had is usually a very collaborative research approach that can we find and work with others to, expand our data sets, expand our network to the point that I think across all the different works that we've had, I think we have about 65 unique co-authors, some of which are on the call today, across probably like 30 or 40 individual research organizations, either as companies or as universities or as regulatory agencies. So really excited to continue on this collaboration and expand our network with this call today. And as mentioned, I have bounced around a couple different places over my time. So if anybody's really interested to have a conversation in broken Spanish or French, I'm more than happy to participate. Moving on, like this is our paper. So you can find it. It's in Water. It's open access. And I've also cited a companion paper. And so, Brett and James and Zahi and others, they had developed this, they have a pilot scale desalination unit where they would develop the test water for us. And then we would do the chemistry, chemical characterization, and then also the toxicity testing in our lab. And so that's why there's this wide representation across all the folks involved in all those different steps. One of our key objectives was to, do as much as we can, measure all the things, do all the toxicity tests, more than we quote unquote would need to. And the idea was we could comprehensively characterize the water in terms of the toxicity and the chemistry, could we then find some more practical tests or assays or surrogates that could be used for more routine applications? And this specific activity is to support beneficial reuse of the treated, desalinated produced water. We'll get into this in a minute, but along the way, like the organizing principle for this work is really based in classic risk assessment. That we have this test water, and we're going to analyze the chemistry in it, and then also do the toxicity tests. But then there's a purposeful step to integrate both of those pieces of information. And I'll step through in some detail how we do the toxicity modeling to integrate those pieces of information. So now we can look at what are the actual relative potencies of all the different things we're looking at, and then how do you add them together? And then there'll be some thoughts on implementation, because some of the findings of this are and would be like, what are the key stressors? And then once you understand the key stressors, you can then, of course, design your treatment systems and or inform the regulatory discussions on where to focus your efforts. And again, I'm happy to receive clarifying questions throughout. And I also think that we'll have a lot of time at the end for some really good Q&A. So let's go. This slide, what this slide is showing is the produced water volumes by year in the Permian region, so the broader Permian region. And really, this is just a talking point or a point of reference to illustrate that there's a lot of water to manage. And this beneficial reuse may have a role to help manage the large volumes of water that we have. More on that in a second. But the water, so this second slide here, this is a slide that I received from our friends at the Pioneer who just joined us last year as part of a acquisition of ExxonMobil, and we're really excited to have them part of the team. And this is one of their slides that they shared with us that sort of illustrate where all the water goes within the operation. That starting way over in the side here, of course, there's the production well, and probably well known to most of us here that we produce quite a bit more water, on the order of 2 to 5 times more water than oil. So there's a relatively substantial, or there's a lot of water that comes out with during the production of the operation. And there's different ways that it's classically managed. So today, most of the options include either recycle or disposal through the saltwater disposal wells. That's what this SWD means. I know that that's a point of focus within the community, within the operators, across the operators, is to find that balance and to optimize where you can to recycle. Because if you can recycle more of the water, of course, then less of it needs to go down to disposal wells. But in addition, there's been quite a bit of effort over the last few years on taking some of this water and then cleaning it or running it through desalination and polishing so that it can be used potentially outside the oil field. And that will open up another avenue for not just to manage the water, but also to supply or meet some of the needs for the water demands in the region. And that's what this next slide represents. This is work by Bridget Scanlon and others at University of Texas, where they are evaluating or balancing or rather illustrating the agriculture demands versus the produced water volumes that are available, and also the fracturing water that's used in the operations. And what this generally is showing is that the green balls represent the irrigation demand, and that in most regions where there's operations like these sort of where produced water is available, there's a pretty substantial irrigation demand. Now, obviously, not all of that will be met with the treated or desalinated produced water, but there is this potential that there is a large demand, and to some degree, with some uses, there's this potential to reuse the water for these other beneficial uses outside the oil field. But to do that, there's a lot of interest in how do you demonstrate that the water is clean and safe for use, and that's where most of this work comes in for the next several slides. This slide here, and I know that we've gone over this, other presenters, and these data are broadly available that demonstrate the composition of the raw water. And I'm showing this for a specific purpose. For when we're doing the toxicity analysis, we're often in a scenario where we either have too much salt, like in the case of the raw water, where we have like 120,000 milligram per liter of total dissolved solids. And then when we get to the distillates and the finished polished distillates, there's a really low ion content. And that becomes really important on how you interpret and conduct the toxicity test at the end. So we'll get there along the way. And what we notice is that usually it's the sodium chloride that is the major ion pairs in this in the produced water, in the raw produced water. And this next slide, this next inset, just represents this is a specific treatment train that we use in our system in our paper. But it represents more of a category that treatment technologies do exist to treat the water, to desalinate the water. There's a variety of thermal techniques, there's a variety of membrane techniques, and others, hybrid techniques and all the different types in between. And this is more a commentary that there are techniques available at both bench scale and a pilot scale to treat the water. And when you treat the water through this desalination process, what happens, of course, is that you can pretty effectively knock off most of the salt. I think most of the field pilots can probably achieve total dissolved solid concentrations under 500 milligram per liter. But of course, the composition of the salts that are left in the distillate change a little bit. And part of that's because you obviously removed most of the sodium and chloride and other things, but you see a little bit of bicarbonate. In this specific case, bicarbonate is one of the major anions in the specific composition in the distillate that we used. Now, what I'm showing here is when folks are doing the toxicity analysis of the treated effluents, what they commonly do is mix the distillate, which has that really low TDS and that specific composition, and they mix it with a formulated recipe water. So usually it's this EPA moderately hard reconstituted water to do the testing. And that's because the test organisms are well acclimated to that, and it represents more or less the receiving environment that we operate in. But it has a different composition. And so what you're noticing in that pie chart is that there's a fair amount of sulfate that's present. And notably, we'll get to this in a second, there's calcium. And notably because in the distillates, we often don't see calcium. And that's important because calcium on its own is an essential nutrient. It has a role in either protecting the organism and or just enabling its function. And so what I'm showing you here, there's a conversation on the side here, but there's also this plot in the middle. So I'll start on the side, is that the nutrient requirements of the daphnids and the types of organisms that we typically use in wet tests, like the Daphnia, typically have a calcium requirements of more than 5 milligrams per liter as total calcium. That's a number that you find in the ecology literature, and that's what the Cairns and Yan paper are. Or you find it when folks who are looking at the osmotic stress or the toxicity of salts in a general sense, and that's where you see the paper from Erickson and others, where at really low calcium, you start to see stresses. And that's also what we empirically see in the toxicity data that we develop for these finished waters. So the chart that I'm showing you here, it's the performance in the Daphnia reproductive inhibition test, just in 100% distillate. And we'll get to this in a minute. I'll show you some of the chemistry later on, is that most of the chemistry is very, very low in TDS, very low in the chemicals, and also very low in ammonia. So mostly what we're observing in this trend is that at really low calcium, when you use the distillate just as received from the desalination units, the calcium is very low. And then you also see that corresponding really low performance in the toxicity tests. And that is interpreted because, and that coincides very nicely with this 5 milligram per liter threshold for essentiality of the calcium. So we began to notice this early on in a lot of the tests that we would run. And so pretty quickly we started to remineralize the test waters. And if you do that, then you get this collection of data in the middle where most of the data seem to perform as well as the controls. And so that any effects that you might be observing are probably due to some other chemistry in water, and not necessarily due to the calcium deficiency. But in some of our other trials, as we try and balance the we're trying to balance the like add back the calcium. Um, Depending on the form of calcium that you use and how careful, like the loadings that you provide, you can in this specific case we overshot and added too much calcium, and so now we're in a range where you have relatively high osmotic pressure because of the chloride counterions. So you can have also toxicity if you're not careful about just how much calcium you add to the system. But this is to reinforce, the idea of like the critical review of the toxicity data themselves, because there's a number of different, stressors or issues like essentiality of calcium that need to be evaluated as you validate those data for the further use. I'm going to move on. So these are maybe some quick slides just to illustrate that the raw water itself can cause some toxicity. These are toxicity due to, this is toxicity of algal growth inhibition exposed to the raw water. And in contrast, as a function of the source water, like as you dilute the water, you see the toxicity go away because you're diluting effectively salts. But of course, there are other stressors involved or available in the raw water, including some hydrocarbons, ammonia, and a couple other things. But once the waters have gone through the desalination system and you have all the pre and post treatments, you see that the toxicity disappears, coincides with all this treatment. Now, this is an example of, and I'll show you more of the data across in our study and across the literature in just a minute. But after the review of the toxicity data and this interesting feature that the Permian is really a situation where you can have too much salt or not enough salt, but of course the right kind of salt. You need a little bit of calcium to support the toxicity test. So the second thing we wanted to focus on, or one of the other things we want to focus on, was a critical review of the chemistry results. Because we and many of the other research groups have been measuring a very long list of all the chemistries that we can think of. And so I think there was a seminary series itself on the NPTES Plus list. And so different iterations of those long lists of chemistry have shown up in the research community as we try and measure everything we can think of. And so I'll comment, though, that as we look through those long data lists of data, there are some chemical classes that we measured because we could, and we felt some responsibility to characterize them, but they're simply not present. And so some of the constituents like dioxins and PCBs, pesticides, herbicides, and chlorinated solvents, they're on the list, they're standard methods. We did look for them, but we did not find them. But so moving forward, those lists are, because they're not used, they're not measured, they're not purposely added to any of the produced water operations, they probably don't need to be on the characterization list moving forward. However, things that we did measure, of course, there's lots of hydrocarbons and some other things. And so there's this table at the top, and this chart is showing more, it's the same actual data on both sides, that there are a fair number of hydrocarbons that are measured in raw water. It's no surprise because the water is in contact with oil. So you measure lots of oil and grease. You measure lots of signature constituents, like very, high ring number pHs or high carbon number aliphatics. So they're very indicative of oil in the raw water. But what's also notable, and of course you also see VOCs in there, and there's also radionuclides, metals, and other things that we measured. But as you start to march through the treatment system, as you go through all the pre-treatments and then the desalination step and the post-treatments, what you pretty quickly observe is that the number of chemicals that you measure drop off pretty fast. This is just the number of analytes. This isn't the concentration yet, but it's a reflection of the efficacy of the treatment systems. But also what this analysis suggests is that there are probably some reasonable surrogates that you can use to characterize the fate of the different classes or categories through treatment. So for example, like the BTEX molecules, they're easy to measure. They're a clear indication of the VOC content of the oil, and they represent more or less the other VOCs that may be present in the neighboring chemical space around them, or other surrogates such as TPH or other things might be useful. And some of that's based on what you find in the finished waters, and then you can look back in raw waters to confirm that. But this sort of analysis, I think, provides a basis for focusing on which sorts of constituents could we routinely measure that represent the performance of the treatment systems. So I'm going to move on now from the chemical analysis, and I'm going to talk about now how we integrate the chemistry data with toxicity data. So the way that we classically do this problem is we use a concept called toxic units. You might also be familiar with relative potency or relative risk or risk quotients. They're all relative, more or less the same sort of approach, where the toxic unit is more or less the sum of the ratios of each constituent. So meaning the measured concentration against its toxicity limit. So I'll talk about these separately. So this measured concentration up here, so I give this table as illustration. These concentrations are for the BTEX constituents plus naphthalene plus phenol. These are the measurements that we have in the polished finished water. So this is the concentration. This becomes this red symbol on top, this CW. But you need to have then the toxicity limit. And because these lists are very long, often there's not empirical toxicity data for every constituent of interest. And then as you get into the toxicity data, pretty quickly you recognize there's a lot of variability within toxicity data and requires some careful quality control, analysis, and screening. And so to address some of that large effort, we've relied on these modeling techniques that are provided above, but also listed in these citations below, as we use these so-called critical body burden models to help fill in the gaps or provide reasonably high quality and defensible risk limits, or not risk limits, but toxicity limits for a long list of constituents. And these models are represented generally in this sort of algebraic term, where you have some LC 50 or an EC 25 or some endpoint of interest, either acute or chronics. And it is a function of the partitioning property of the molecule to the test organism. Very often, this K biota, this partition coefficient to the test organism, is often represented in some form using a log KW or some other modeling techniques, which is sensitive to which represents the range of the physchem properties of the molecules, because some of them, of course, are more sorptive than others, and you see that show up in the toxicity data, where you see nice inverse relationships between the LC50s and, say, the log KW. Those are features that are found in the links in these papers below. But this partitioning phenomenon is balanced by the relative sensitivity of your specific endpoint or organism. And so what was called a critical body burden. What it represents is an internal dose inside the test animal of how much material accumulated in the animal. And then you empirically determine this number by looking at all of the toxicity data. So this becomes a linear regression calculation where someone compiles the toxicity data, and then you estimate the partition coefficients, and then you estimate this critical body burden. And when you do that, you then establish the ranges of the relative sensitivity for both acute and chronic endpoints. I'm focusing on the organics in this specific case, but there are very similar modeling frameworks available for many of the inorganics that we look at. We can talk about that in more detail if there's interest later. But the way that this calculation works in practice, of course, is now for this list, we've calculated these EC25s based on a critical body burden that's based on empirical data. And now we can compare that to measured concentrations, and we can calculate the risk quotients, or this toxic units. Now, what you're observing is that for many of these constituents, they're below the detection limits and below the quantification limits. So simply, they're always less than numbers because it's a really, really low number. And perhaps for a different conversation, a different day, is how you treat the non-detects. But for this specific study, we chose to ignore all of the non-detects as zero unless they were close to some risk limit that needed evaluation. So in practice, where we applied this, as I'm showing this chart here, which is showing, it shows a couple different data types here. This specific plot is showing this sort of toxic unit analysis is applied to the longer list of hydrocarbons specifically. So they're indexed by their log KW. So the log KW is a measure of how big the molecule is. The larger molecules with the more rings, of course, have a higher partition coefficient, and that's what this represents. And so that's why you get this molecule way over here. You get toluene is listed. It's this little, orange box. There's just a tiny amount of toluene that was measured in the finished water. And then there's a tiny amount. This is naphthalene. That's what this other square represents. And as you continue to go along, you start to see some of the higher ring constituents present. But notably, most of the measurements are below detection limits. So they're estimated in some degree. There may be some small peak on the analytical, but they are below the detection limits. And the other piece of information to consider is like the chronic toxicity threshold. So that's the information we just derived in the previous slide, is using those modeling techniques, you can calculate the empirical, either acute or chronic toxicity thresholds for these chemicals for the test organisms of interest. And what you're observing is that there's a really substantial gap on the order of factors of 10 to 100 or more between the measured chemistries and the toxicity thresholds. And at the end, we'll talk about some of the other non-targeted analysis or some of the integrated passive sampling work that we do, which confirms this result, that most of the organics have a pretty low contribution in these specific tests, in these specific waters. And this becomes important because as you look at the next slide, this is the summary for some of the notable inorganics. It's organized in a similar way that there are, so in this specific collection of data here, this is the total dissolved salts. This diamond is the toxicity benchmark for TDS as a whole, as a milligram per liter of TDS. This red box is the measured TDS concentration in a finished desalinated water. It's a pretty low number. This circle then still represents the detection limit. So in this specific case, the interpretation is the salt is not a toxic driver in the finished water. I'll skip over to this side because zinc was also a notable measurement that we didn't have a quantifiable measurement for zinc, which is down here. It's this red box. But the environmental quality standard for zinc is around 100 microgram per liter, which of course is greater than the measurement. So zinc is probably not the driver for the toxicity that we'll talk about in a few minutes. But what does become really noticeable is the ammonia, because the ammonia is the only constituent that was measured at this measured concentration, this red concentration here on an order of about 20 milligram per liter, that was above the risk limits of 2 milligram per liter. And also a detectable number. So ammonia stands out as in this specific data set as the only constituent that was above the risk, the toxicity thresholds. And this toxicity threshold, I'll comment a little bit more. Ammonia is a relatively well-studied constituent. There's some really nice, like the EPA water quality criteria is a very nice document that compiles lots of tox data for freshwater organisms, and then evaluates them in terms of its pH dependency, which is important because the toxicity of ammonia is pH dependent. The pKa of ammonia is at 9.2. which means below 9.2, you start to add the protons, and then it becomes a positively charged molecule. And of course, everything happens in logarithmic space with the pH. So if you're testing, like you're in your wet tests, if you're running your wet test at a pH of 8.2, say, that means only 10% plus or minus of your ammonia is in the unionized form, or rather the toxic form. That becomes an important point of interpretation, is that when you run your toxicity test, what is the pH of the test? And that's important for interpreting the wet test, obviously, because if the water itself coming out of the distillation in the polishing step will have some pH based on the chemistry of that system, but in the wet test itself, it may have a different pH. And I think very often in some of the testing labs, they normalize the pH so that it's not a confounding factor in the tests, or they adjust the pH to a common value of around 8 to 8.5. So ammonia stands out as a really clear stressor. So I'll show you some of the data and how we interpret that in a minute. But I want to demonstrate this first to go through this exercise. Because what we're doing here is we're applying this approach, like this toxic unit modeling approach, to the measured chemistries across the treatment train. And this is a helpful exercise because it helps focus on, like, is the treatment system working the way it should be working? What should we be removing? And in the raw water, there's this pie chart up here. There's a lot of toxicity in the raw water, no surprise. And the toxicity is driven more or less in equal parts by the organics, mostly the hydrocarbons, and the TDS. There's a lot of salt there, and the ammonia. So the ammonia in the raw water out of the formation is, on the order of 400 milligram per liter. It's a lot of ammonia there. But as you start to go through the treatment systems where you remove the organics, where you start to remove the other constituents, and certainly once you get to the desalination step, you're removed the TDS and most of the organic constituents. And certainly once you get to the polishing step where you purposely remove the ammonia, you see the toxic units drop off really substantially. So I think this is a helpful insight to focus on what should we remove. And fortunately, ammonia removal is a relatively well-established technology, and there's a number of different technologies to do that. But that's really helpful because now that we understand what the stressor is, we know how to manage that. So I'll show in a minute, this is the toxicity data. And we can get to this. I'll pull this up in the next slide and we'll walk through this in more detail. But what it's showing is the effect in all of these different wet tests for all these different organism types that we tested as a function of the unionized ammonia concentration. Remember, it's pH dependent. And what we're seeing is that in our specific test, the test data that we derived are these colored points. So that's a Cereodaphne mortality, that's a green algae growth inhibition, and there's rainbow trout, or not rainbow trout, that's zebrafish in here too. And there's also these little stars, these little pink stars, which represent cytotoxicity and some of the cell line testing that was done. And what we are observing is that the toxicity where there is toxicity happens above this toxicity limit that was established in the EPA criteria document. So it becomes another line of evidence for us to focus on ammonia, that where we see the effects, it's where we have high ammonia. I'll point out there's a lot of like open gray symbols in here. These are literature data analyzed in a very similar way, and it has a very similar result, that where you have the high ammonia concentrations above the risk limit is where you start to see the toxicity. Now, you're probably also noting there's some variability observed in the data sets. I'll note specifically like some of these zebrafish data. In our test with our lab, with our strain of organism, the zebrafish was not very sensitive to the ammonia. And then some of the other data types and some of the other test organisms, very similar response. Some organisms are just not as sensitive to ammonia as other ones, which is consistent because if you look at the EPA criteria document, there is a relatively wide range of sensitivities across organisms. with this toxicity limit representing more or less the lower bound. So maybe I'll pick this apart a little bit more. So on this next slide, this upper panel here is the same. It's the same data we just looked at on the previous slide. These are all the acute tox data. So there's Daphne, or there's green algae in here, all the circles, the Daphne mortalities, and there's the cytotoxicity. There's also microtox represented, earthworms in here, and also this is a seedling germination assay. And all of these different assays for the acute limit show more or less the same story that we just talked through. I guess I'll note real quick, the green algae test is sometimes difficult to run in a wet test assay. Like you can run the actual test, but the interpretation can be difficult because what you sometimes observe is that you have like a slight enrichment in the algal growth, probably because of the nutrients like the trace amounts of ammonia that are there. So the algal test is I mean, we have some interesting data that seem consistent with the other data, but it's often not probably the most stable test assay. The test assays like the Daphne and the fathead minnows that we'll get to later seem to be pretty stable assays for these systems and reasonably are sensitive. So, and that's like partly what this lower pot is getting towards, because these are some of the chronic endpoints. So we tested chronic endpoints for the Daphnia. So this is the reproduction assay where you run it out for 6 or 7 days, and the number of neonates per female. And of all the chronic long-term tests that we ran, this is the most sensitive test that we ran. That's true for both the earthworm test that we have, the fathead minnow, not the fathead minnow, this is zebrafish, where we're looking at zebrafish deformities. And also, like, it's sort of buried in here, but you see that there are 2 symbols down here for different plant types, like this Brassica rapa and this Avena sativa. They're 2 different ISO standard toxicity tests, but they're terrestrial tests. So you're growing these plants in soil for up to 3 months, where they're growing and eventually forming seed pods and flowers. And what you're observing in the one with the sativa and specifically is you see a slight enrichment in the biomass in the flowering indices, similar to what you saw with the algae. They're probably responding to the addition of the ammonia that's there. But the conclusion that we draw from this is that ammonia continues to seem to be the major stressor in these systems. And the Daphne specifically seems to be a pretty stable and sensitive assay for characterizing these systems. And this is the data set, more or less the data set that completes our paper. So the next thing I'm going to show you is some like future work, because we continue to try and compile data across wherever we find them, across all the, opportunistically in literature or through some of the project work that we're involved with and some of our other partners. So this represents some of those initial data that we've been compiling. And, as I'm sure you're all aware, there's quite some few, pilot data coming online. And so it's going to be really interesting next year or two as we see all these data sets come out in literature. But all of these studies are, if we focus on panel A, it's very similar. In this specific case, it's just a summary. We've pooled all of the data together for all the different organism types. So that does contribute to some of this messiness back here. But it does allow us to make 2 different points here. One is that we've color-coded these systems in between, very generally speaking, there are membrane-based systems and thermal systems. Now, I think in actual practice, there's probably like 4 or 5, or no, there's like 6 or 7 different actual technologies in this data set. But in a general sense, we're looking at, is membrane different than thermal? So by colors. So the short answer is no. We don't see strong differences between the different treatment systems, which is really encouraging because what it indicates is we can achieve clean water in all the different test systems. But what this specific data set includes is toxicity data from the distillates themselves before polishing as well as some data after polishing with a specific removal step. And that's what panel B represents. Panel B represents the subset of that data that had the purposeful, deliberate ammonia removal steps. And as you remove the ammonia, you see the decline in toxicity, which is, again, another line of evidence to reinforce or confirm that observation that ammonia seems to be the major stressor. Now, you will also notice there is a healthy amount of variability in these data in this broad data set, including a couple points that bounce up top. Some of that represents noise in the biology. Some of those are real results. Like in this specific case, there's this this blue circle at the top, or this brown circle at the top. The ammonia concentration is not zero. There was an attempt to remove the ammonia, but it didn't push it down far enough. But in general sense, after you remove the ammonia, you see also the decline in toxicity. I think the last thing I'm going to spend a couple minutes on is this use of our passive sampling technique as a way to measure the toxic potency of the organics, quote unquote. It is a form of non-targeted analysis, but instead of trying to analyze and characterize all the peaks in the mass spec, our goal is to measure everything on this fiber, on this passive, this solid phase microextraction fiber, and use that as a proxy for the bioavailable chemicals that a test organism would experience. So where this starts is this is work that Tom Parkerton started many years ago, and we've since built a relatively substantial validation data set in classic laboratory tests, but also in some field data sets as well. It uses existing technologies, including SPME and these GCFID and other analytical techniques, where you take this fiber, it's coated with a certain resin, a PDMS. I forget the acronym right now, but the PDMS is a relatively common polymer used in these SPME fibers to extract and concentrate the chemicals. And the research community use these techniques very broadly to extract and concentrate chemicals for analysis in all kinds of waters or test media. Our specific nuance within this world of passive sampling is to use this in a non-depletive manner. For the organic chemists out there, our goal is not to extract every molecule from water. So much like a test organism in your beaker of water, you have a small test organism. It equilibrates with your test system without removing all the contaminants. It's a very similar principle. We're into the sense that this little passive sample or fiber, which is represented in this little box here, is therefore a proxy for the test organism in your system, either for the Cereodaphnia or the fathead minnow or the Daphnia or whatever animal, test animal you have. So once you equilibrate your sample with the fiber goes in your GCFID, and it goes through a relatively quick ramping phase where your idea is to express or elute all the chemicals pretty rapidly for measurement by FID. Again, we're not trying to measure peaks. We're trying to measure area under the curve. That area under the curve then becomes a proxy for the body burden, the exposure that the animal experiences during the test. It's not actually the same thing, but it's a reasonable proxy because the sorts of processes that control the partitioning to the fiber are very similar to the types of processes that control the partitioning to the test animal. And as sort of a proof of concept, I give you this plot in the middle, where all of these tox data, this is mortality for, in this case, I think it's mysid, against the amount of oil that's added to this test system, like this beaker of water, for a number of different substances. And so you see a very wide range in responses. And this reflects the different solubility properties of the different oils that we're looking at. But then if you re-express that same actual toxicity data in terms of the measurements on the fiber, on this BESPME measurement, you see everything collapse. And now the toxicity data, like there's some noise in there, but that's not very different than other well-curated toxicity data sets. And you can see the variation drops from like 3 orders of magnitude to like factors of 2. And so as a practical method for measuring the toxic potency of really complex mixtures, it's a really promising tool to fit in this niche. So we thought we should apply this here too, because with the produced waters, we have a very similar scenario. Like there is a mixture of chemistries, some of which we know and some we may not know. And can this technique help us interpret that? The other piece of information I will add here is that we've done this like toxicity analysis, like these dose response analysis for lots of different chemistries and lots of different organisms that are documented in this list of papers here, to the point that we can estimate, either LC50s or EC25s for some relatively long list or some list of test organisms for both acute and chronic endpoints. So now we have some understanding of what the empirical toxicity benchmark should be. And that's what this last slide represents. Um, this last slide this is the measurements, or rather, this blue bar represents the compilations from all of our literature work before. Like these are the thresholds for acute toxicity across a range of different organisms. And it has like these units of around 300 or so, with some range, and the units are millimoles of material that absorb to the fiber. We've also done a similar exercise for the chronic toxicity data, and of course it's offset, it's lower as normally expected, but we've now established more or less the range of what we might expect for chronic toxicity. Now, as we apply this to the treated waters, including the study that we just talked about, including some of the other pilot study that are ongoing, we see that generally speaking, these green measurements are really low. They're typically this is a logarithmic scale, but they're typically on the order of 2 as an arithmetic number, um, which is well below the acute and the chronic effect thresholds. And it's consistent with the toxicity modeling that was done before, which showed that it was really low potency from the organics. And again, it's another line of evidence to help confirm that result that we're finding for ammonia. And so maybe with that, I'll sum up here, is that the pre and post treatment around the desal methods are very effective at cleaning water. You find really substantial removals. It's not the focus of our paper, but it's a clear, obvious feature of those systems. Ammonia does have some transference to the system, probably because of its moderate volatility or other transfer mechanisms. But as you polish out the ammonia, if you remove the ammonia, that coincides with the decline in toxicity. And with this analysis, it helps us focus in on what constituents where our attention should be placed. So specifically on ammonia removal. Obviously, along the way, we're removing the organics, which is a very important step also, and the salts and the metals and other things. But in the finished water, I think we have some understanding now of where we should focus our attention. And so I think with that, I think that's about the end of my discussion. So I'm happy to take questions or comments or.
Very cool. Well, thanks, Aaron. Thanks for your time and for the amazing presentation.